So this is fun. I used to do like 8,000 of these. And I'm going to put you guys to work today, for sure. So I want you guys to try some stuff out for me. So there are two different ways you could run this test. You can do it to detect antigen, or you can use it to detect antibodies. Antibodies. Wasn't that the bonus question today?
No.
It was. It was. Right? All right. So sometimes referred to as the indirect ELISA, where the direct ELISA is directly detecting for antigen. So ELISA is actually an acronym. It stands for Enzyme-Linked Immunosorbent Assay, right? So we're using an antibody in the detection. Even if we're looking for an antigen or an antibody, we use these special types of antibodies in this test. They're special in that the antibody has an enzyme. Yeah, this is not Pac-Man, but it's to represent an enzyme that is linked to an antibody, right? So this is the antibody. This is our little stick drawing for our antibody. So as the name says, enzyme-linked, immuno referring to an antibody, right? An antibody is a product of your immune system. Sorbent referring to a color change. Assay referring to a series of steps. Can you do these in any order you want? No. If you do not do them in the correct order, it will not work. So an antibody is a product of your immune system made by plasma cells, which are derived from B cells or B lymphocytes. And they're specific to the antigens they bind to. And what an antigen is a foreign substance that you can make antibodies against. All right, that's one of the most common things. So we actually use antibodies to detect antibodies, right? Because for one antibody, it can be an antigen for another antibody. So when you say antibody, oh my God, right? You're like, ah! What is she talking about? Right? So a lot of times we'll refer to it as an immunoglobulin antibody. And what an immunoglobulin is again another synonym for an antibody. But what this is an antibody that binds to another antibody. And that's what our secondary antibody, the one with the enzyme linked to it, is. Right? It is an antibody that binds to antibodies. Hopefully, going through it'll make sense. Okay, so again, little stick drawing of an antibody. The general structure of an antibody, it has two proteins that are identical, that are really long. They're called the heavy chains because they're bigger. And then we have two light chains. And so the very simplest structure here of an antibody is two light chains and two heavy chains. At the tips where the heavy chains and light chains are, is what is called the antigen binding site. So this is actually where the antibody binds to the antigen. So at this A and B site, what's going to happen is this antibody is going to attach to these antigens, right, in our little wells that we're using today. This portion of the antibody, known as the FC region, is where the secondary antibody is going to bind, right? So where could an antibody bind to an antibody? Down here. So we're going to be like stacking things in our wells today. We're going to start out with an antigen, then we're going to add an antibody, and then we're going to add another antibody on top of it. So we're going to create like an antibody sandwich. So in your blood, right, the serum, which is the liquid phase of your blood when your blood is allowed to clot, you have lots of different antibodies to bind to different things. So that's why these are represented in different colors, to denote that each one of these antibodies is different and binds to a different antigen, right? So different foreign substances. So why are they different colors? Because they are different, right? And there are different classes of antibodies. We're not going to get into that in this class, but in your lecture classes you should, and we will. So you may have heard of them. They're commonly abbreviated IG for immunoglobulin, which again is another name for an antibody, and then a letter denoting which class of antibody it is that it's used. So again, remember that specific region at the top, that chain is different for each antibody. That's why these guys are different, is actually what binds to the antigens. This portion interacts with the immune system in our bodies. In our case, it's going to be where the other antibody binds. So plasma is when that liquid phase of your blood, when we take blood out of your body, there has to be an anticoagulant in that to stop your blood from clotting. As an immunologist, I was not too happy when my boss would give me plasma, because plasma has all the clotting proteins still in the liquid phase of the blood. And that would interfere sometimes with these tests. I preferred to use serum where the blood was allowed to clot, right? And then that liquid phase you're left with that contains the antibodies just like plasma does, but it doesn't contain any of the clotting proteins because they're in the clot, right? So that's the distinctive difference. Tests can be run on both types of the liquid phase of your blood. The antibodies are there. Just, we usually prefer serum because it doesn't have the clotting proteins.
So when you have like a droplet of blood and it clots, all of the clotting properties have like drawn to the...
Yeah, most of them are in the clot, yeah, that are in the liquid phase. So serological tests actually get their name from the fact that they're usually run on a serum sample, that liquid phase of your blood. And again, we're looking for things that are in that liquid phase, like antibodies and even antigens can be found in the liquid phase of your blood. So antibody or antigen really depends on what you're trying to find out, right? So for us, we are doing the antibody detection, which is also referred to as indirect, where antigen is referred to as a direct ELISA. So Bio-Rad, the company we buy these from, actually have a pretty cool animation that I put the link for you guys in Canvas. I'll warn you though. So here they are. No audio, right? Oh, I don't know why. I can just link it out. Oh, I gotta allow my flash. Activate. I've hit allow and remember I don't know how many times, but it doesn't matter, yeah. So they have both of them, right? Antigen and antibody. So you can look at both of these and compare, and you see how it flashes. You have to keep hitting this button, and it goes through the steps, right? So we're going to add purified antigen to the wells. We're going to rinse out anything that doesn't bind. We're going to wash it out with wash buffer. We're going to add those antibodies, right, where it could be a whole bunch of different ones from a serum sample from a patient. We're working with purified antibodies today, not anything from a patient, not anything that's going to hurt you. It's just proteins. We don't have to wear gloves or goggles or any of that crap today. And all the things that we use that are disposable can go in the regular trash can. Yay. Right? So it's all purified stuff. It's not from patients or anything like that. It's a kit. So the one that binds to the antigen is going to do that. You see that there? It's going to actually bind. Any of the ones that don't bind, we're going to wash out in our wash steps. Right? So bye-bye, right? Only the ones that are attaching will actually stay behind. Then we're going to add the special antibody that binds to antibodies that has the enzyme linked to it. Right? So this is what makes it an ELISA test. And notice it's binding to the antibody, not the antigen. Right? So it's helping us detect that this antibody to the antigen is there, that we're looking for. Anybody who doesn't stick, we're going to wash out. Well, these are proteins, right? These are like submicroscopic. You're going to see any of these in any of your wells? No. So we need something to see, right? So we're going to add a substrate that's pretty much colorless. But when you add it to this, that enzyme, that little Pac-Man, is going to cause an enzymatic change in that substrate, and it's going to turn, anyone know? Blue. Right? It's going to turn blue. So as we add each one of these reagents, we got to let it sit for about 5 minutes before you guys can dump it out, right? And then rinse. So, although lots of cool information. So when do we use these types of things? So the rapid strep test, guess what? That's an ELISA-based test looking for the antigens, the bacteria that cause strep throat, right? It's a little snap kit test, right? Pregnancy test, right? The antigen is the hormone you overproduce when you're pregnant, right? Ovulation test, that hormone again is the antigen. We're using antibodies to detect it. Drug tests, HIV antigen testing, rapid flu tests. These are all ELISA-based rapid tests that they use in doctors' offices or at home. Antibody ones are when we're specifically looking for antibodies. Do I have examples of those? Yeah. So when we're looking for the antibodies someone may have produced against HIV, for your pets, right, when they're taking that blood sample and they want to know if your dog or cat has heartworm, they're actually looking for the antibodies that they produce against the heartworm usually with those kits. For hepatitis viruses, they're again looking for the antibodies you've produced against them in a response to that infection. So I'm not going to talk about HIV. I'm going to skip over that. So this is just a diagram, just like the animation, right? It's going to be antigen. We're going to stick to the surface. Then the secondary antibody. First antibody, primary, secondary, which has the enzyme. Add the substrate. We get the color change, right? Other ones will use things that produce fluorescent products, right? Or even chemoluminescent products, right? There are different things, different types of enzymes they can attach, right? In the case of ours, it's just a pretty color product. So this is an example of where they've used antibodies that have been tagged with fluorescent dyes to stain slides. And so the antibodies are binding to specific things in those cells. So you'll see that the protein they were looking at, right, this green one is stretched out throughout these cells on the inside. You can see this red one, right, is pretty diffuse as well. This one that they coded green, look at this one. It's really associated with the membrane in these cells, right? So it's pretty amazing stuff that we can do with antibodies because they are so specific as to what they bind to, right? And we can add stuff to them, right? So that we can help visualize and see these things. So, beautiful stuff. So, for our procedure, right, it's pretty step by step, right? Hopefully, let's just make sure you guys have your steps filled in correctly, or you have your lab manual. So you're going to start with a little tube that contains purified antigen. It's a green tube. And we're going to add it to all the wells, and you're going to incubate it for 5 minutes. Then I'm going to show you guys how we're going to wash it out. We're going to shake it out onto a stack of paper towels. And then we're going to use a wash. And anything that doesn't stick is going to be removed, right? Why do we want to remove stuff that isn't sticking? It doesn't interfere with our test, right? We don't want anything that's not binding, right, still there. We're not going to use the, there's one step in there. I haven't got to it yet. We're going to wash twice, right, for the first wash. So you're going to do it twice. Why 2 washes? Because again, we're removing things that are unbound. What happens if you wash too much though? We're going to remove our stuff that we need, right? In simplest terms, right? We don't want to do that. But if we don't wash enough, right, we're not going to remove the stuff that didn't stick, right? And this will interfere with our test results. We're going to skip, and I think I have it in your protocol, right? We're not going to do the blocking agent. Our buffer that we're using works pretty good as a buffer. Although we're going to find out today if it'll work on other plates. The next step is what's important because each one of the wells that are labeled, you add a different reagent. So we're going to do a positive control. So it definitely has the antibody in this run, right? To all the plus wells. And we're going to do this in triplicate. So you're going to add it to 3 wells. Then you have to change your tip, right, to do your negative. So why do we do positive controls anyways? So we know what positive looks like, and we know in this case that we performed the test properly and the test is working. So in those pregnancy tests you can buy at home, right? You can do at home. It has 2 lines, even the ones with the digital readouts, right? If you remove it, it has 2 lines if you're pregnant. One of them is your test sample. What's the other one? The positive control. If the positive control doesn't work, what does it tell you? Error. Repeat test. Right? It's not good. So in this case, right, we know that the primary antibody is there. Right? That's what we're testing for in this test, is that primary antibody. So we want to ensure it's working properly. What happens if we get negative results in the positive controls? Can we use the results for our unknowns? No, it invalidates it, right? Something went wrong. We may have watched too much, right? We might have added the wrong reagents. The reagents might be expired, which they were. I tested them this morning. I had to reconstitute all new reagents for you guys this morning. So it damn well better work. So positive controls, this should happen, right? But we're not going to see it. But this is what should happen. The antibody should bind to the antigen that we stuck in the wells. Negative controls, again, this doesn't contain the antibody, right? No primary antibody. And this is to show that only the primary antibody will cause a positive result, right? We don't really have that issue with pregnancy tests and those types of tests. They don't really run negative controls. But for these types of tests in a lab, you would definitely run a set of negative controls. Right, so we want to make sure that it's just that primary that causes this reaction. What would cause a positive result in our negative controls? Cross-contamination, right? Failure to wash out stuff, right? If you didn't wash out the antibody and we put the next one in. Contamination, I'm going to show you guys how to do that so you don't contaminate. Would positive results and all negative wells affect your interpretation of results? Absolutely, we invalidate them, right? We don't want that. What would a negative control look like? So we have antigen in these wells. What's not going to be here? The antibodies, right? No antibodies, right? So notice no antibodies attaching. So the other wells are going to be numbered for your patient samples. Patient number 3 and patient number 4. Obviously, one of them is positive and one of them is negative. Your guys' challenge is to figure out and do it properly so you can find out who's getting positive results, right? Who's getting the good or bad news depending on what you're pretending you're testing for today. I don't care. Why do we do it in triplicate, though? Right. More accuracy and precision, right, if you do it 3 times, okay? And those are important, right? This is very low accuracy and precision, right? We want to get the bullseye and they didn't get anywhere near it, right? Even though they did it 3 times. Where here, they had quite good precision, right? They're all together, but their accuracy is really off, right? Here, high accuracy, but not very good precision, right? They're all in the same area, right? But they didn't hit the target. This is what we want. We want to hit the target with all 3, right? In order to have a nice validated result. For you guys, you're new at this, right? So you may or may not have good accuracy and precision. It comes with technique. Although this is a relatively simple technique. As long as you follow the instructions, we should be pretty good. So poor technique could be the issue, but also poor sensitivity, right? If you don't have enough of the sample in there to create the color change that we need to be able to visualize it. This is a highly specific test, though. We usually don't have issues with specificity. So in a quantitative assay, or qualitative, excuse me, we're asking yes or no, right? Pregnancy tests, things like that. Most of the time we want to know just is it positive, right? And is it negative? Does it have it or does it not? But when you're positive, could you have more of it or less antibodies, right? So for some tests, we want it to be quantitative. We actually want to know how much is there. And since it's a color change that actually can be quantified with a spectrophotometer, there are special photometers. Unlike the one we use where we just put a single tube in, there are ones where you can stick a whole plate in and it reads each individual well. And it tells you a number for the amount of color that's in that well. Right? So you see how this plate varies in color? That's different degrees of positive. And even for this test, sometimes you'll get a little bit of color, and it still wouldn't be a number that we would consider positive, right? Depending on what your parameters are for positive versus negative. So for instance, when I worked in research, and we infected armadillos with leprosy on purpose, right, to grow the bacteria, I would take serum samples if I could, right, if my boss would give me 2 tubes. I would take a serum sample and I would run this test just like you guys are doing, but I would use mycobacteria, leprae antigens in my wells, and I would use the antibodies from the armadillos, and their sample would attach. I'd use a second antibody with an enzyme attached and add the substrate, and I would get color changes that I would read. And for us, a positive result, I think, was it had to be like 700 or higher, based on the control standard we would run. So we would have a control, and notice this control goes from really light to really dark. That's because it's a serial dilution, right? So I knew how much I put in each one of those wells. I could plug it into the computer. Based on these values, these color changes, I could put it in and it would create a graph for me. And then I could put in my unknown number, and it could tell me where it fell as compared to the control on that sheet. And for us, I think, like I said, I think the cutoff was like 600, 700 was considered positive result. But we record both, right? The number and whether it was positive and negative. So before we're going to wash twice again before we add the secondary. So if the antibodies are there, anything that's not bound is going to be washed away. Again, why wouldn't we want to wash too much? We don't want to remove those antibodies, right? We want the ones that are actually binding to stay behind, right? If we don't wash enough, though, ones that aren't bound would stay behind and give us false positive results, right? So we want them gone. The last, or next to last step, is adding that very special antibody, that secondary antibody, that binds to the primary antibody. So what would a negative one look like? At this point, what should only be in the well? Just the antigens, right? No antibodies, right? So is there anything there for the secondary to bind to? No, because the secondary doesn't bind to antigen, does it? It binds to antibody, because that's what we're trying to detect, right? We want to know if the antibody to this specific antigen we're interested in is there, right? So we're creating that antibody sandwich. We incubate, right? Once we add, we're gonna wash out, right? So what does the secondary antibody bind to? The primary antibody, the first antibody. Negative again, just antigen, right? Nothing for the secondary to bind to. Primary isn't there. We're going to wash 3 times the last time. The substrate is light sensitive, so I will give it to you once you've done your 3 washes, right? I will ask you, have you done your 3 washes? Then you can have your substrate, right? Once you add the substrate, literally within seconds, we'll get a color change. Again, we won't wash too much, but we need to wash enough. So how many times? 3 times. Okay, and the substrate is going to be an orange tube. Like I said, I'm going to give it to you. We're not going to have to wait 5 minutes. These are fresh. It'll be within seconds. Once you add the substrate, you'll get your color change. That's it, right? You get your results. Right? So we wouldn't let it go longer than 5 minutes, but it'll be seconds. Okay. Okay, so the actual reagents we're using, we're using a chicken antibody, and then they stuck those chicken antibodies into a rabbit. So a rabbit actually made antibodies against the chicken, and then they took the rabbit antibodies and they put them into a goat. So we're using goat anti-rabbit that actually has the conjugated added linked enzyme to it. And the enzyme that we are using is TMB, and with hydrogen peroxide, and it will convert, the enzyme will convert it into its color form of TMB, which is blue. And so if we read it on the spectrophotometer, we make sure we use the blue filter, which is 650 nanometers for that one. So not messing with these guys today. I don't know why he has that on there. So this is our important key, which is also in the bins, right, to tell you what colors correspond. And I wrote plus and minus on our blue tubes, and of course I had to write the 3 and 4 on our yellow tubes. But green is first, right? I like that they picked those colors too, right? Green is first, pink is last, right? That's our secondary antibody, and orange is the very last tube that I will give you for our test. So let me see